The competent cells have the ability to be transformed, that is, to acquire naked DNA from the extracellular medium to the bacteria by means of an introduced plasmid. To achieve said competence, the chemical transformation or production of chemiocompetent cells by membrane permeability is carried out. Once transformed, it is sought to measure the transformation efficiency by quantifying CFU formed in the medium.
The terms transformation and transfection have always been a source of confusion, because the term transformation was introduced in parallel to define modifications in bacteria and proliferation abilities in eukaryotic cells. So to prevent confusion, the term transfection, derived from transformation and infection, was used to define the infection of cells by DNA isolated from a virus. Finally, transfection is analogous to bacterial transformation, but in eukaryotes, and transformation is used to describe changes in cell cultures caused by viral tumors. The transfection methods are broadly classified into three groups: biological, chemical, and physical. Latter ones are the most recent methods and use diverse physical tools to deliver nucleic acids. The methods include direct microinjection, ballistic particle delivery, laser-based transfection, electroporation, and sonoporation. For bacterial transformation, the method of choice depends on the type of cell: intact bacteria, bacterial protoplasts, spheroblasts, the instrumentation available, the efficiency obtained, and the volume of sample needed. The most used methods to induce the transformations are chemistry, electroporation, sonoporation, and alteration of tribes. However, there is another method, like the Toshida effect. In this method, when a colloidal solution containing nanosized acicular material and bacterial cells is stimulated by sliding friction at the interface between hydrogel and an interface-forming material, the frictional coefficient increases rapidly, and the nanosized acicular material and bacterial cells form a chestnut bur-shaped complex. This complex increases in size and penetrates the bacterial cells, thereby forming a penetration intermediate due to the driving force derived from the sliding friction.
Prior to the development of the practice, E. coli was inoculated on a plate with LV medium and incubated overnight. Then it was inoculated in 5 milliliters of soft medium and incubated overnight as well. For the practice, 1 milliliter of the seed culture was seeded in 100 milliliters of LV medium. This practice was divided into two sessions. In the first one, was the preparation of the competent cells, and in the second one was the transformation. For the chemical competent cells, the culture medium was centrifuged and the supernatant was removed. Then the pellet was resuspended in a calcium chloride solution and incubated on ice for 10 minutes. The same procedure was performed for the electrocompetent cells, but the resuspension was in 10% cold glycerol. The mixture was centrifuged again. The supernatant was removed. For the chemical competent cells, it was resuspended in 2 milliliters of a solution of calcium chloride and glycerol. The electrocompetent cells were resuspended in 2 milliliters of glycerol. It was incubated on ice during 10 minutes, and a liquid of 200 microliters were made and stored at minus 20 Celsius degree. For the chemical competent cells, the culture medium was centrifuged and the supernatant was removed. Then the pellet was resuspended in a calcium chloride solution and incubated on ice for 10 minutes. The same procedure was performed for the electrocompetent cells, but the resuspension was in 10% cold glycerol. The mixture was centrifuged again. The supernatant was removed. For the chemical competent cells, it was resuspended in 2 milliliters of a solution of calcium chloride and glycerol. The electrocompetent cells were resuspended in 2 milliliters of glycerol. It was incubated on ice during 10 minutes, and a liquid of 200 microliters were made and stored at minus 20 Celsius degree. Later, it was taken to the electroporation chamber, which was programmed to 2500 volts for 5 milliseconds. After the electric pulse, 1400 microliters of soak medium was added and decanted to a microtube. In a microtube, 40 microliters of competent cells and 2 microliters of plasmid were mixed and incubated on ice for 10 minutes. Cells were placed in the thermoblock for the thermal shock at 42 Celsius degree for 45 seconds, then cells placed on ice for 5 minutes. 1400 microliters of soak medium was added after incubation on ice. After the addition of the soak medium, both cells were incubated at 37 Celsius degree with shaking for 30 minutes. 20 microliters of arabinose with 100 microliters of cells were spatulated on a plate with LV medium. It was incubated for 16 hours at 37 Celsius degree. Finally, the plates were observed to count the colonies and were photographed.
When visualizing the plates after approximately 12 hours, as we can see in Figure 1, the growth of colonies was observed after the transformation by electroporation, the picture of the left, and by the chemical method with temperature, the picture of the right. In both plates, the growth of colonies was high, so it was difficult to count the CFU to obtain the efficiency, since these were considered countless. However, when visualizing growth, it can be said that it was efficient. In this case, the method that had the highest cellular growth was the chemical, since CFUs are not distinguished, but growth is visualized as a homogeneous layer of cells. In the case of the electroporation method, the growth was also high. However, the colonies could be differentiated. When not being able to count the UFC, we cannot know how good the obtained efficiency was. However, it's known that to be considered good, it must at least be from 10 to at least 6. Reason why it's believed that according to the results could have achieved this efficiency or a little lower. An efficiency down from 10 at least 6 is not considered good.
Calcium salts are one of the most utilized when performing chemical transformation and yield good efficiency for routine procedures and non-complicated DNA. Calcium salts act by neutralizing charges of phosphate groups in double-stranded DNA and in phospholipid bilayers. When applying a heat shock, the induced permeability of the bacterial membrane, aided by the salt charge neutralizing effect, allow foreign plasmid material to enter the cell. Only the bacterial cells prepared correctly will be able to acquire foreign genetic material, which can be measured as an efficiency. Regarding the DNA sample, a high purity is needed, but it is not as strict as for the electrocompetent cells, because this method comprises an electric pulse to briefly generate pores whose surface is very hydrophilic, allows polar molecules to permeate the cell. If the DNA sample or the bacterial transformation suspension contains salts, the conductivity of the medium increases and the cells may die. More colonies were observed in the plate corresponding to chemocompetent cells, Nevertheless, this statement is only qualitative, as both plates exhibit an uncountable number of colonies. If the colonies were sharp enough to count, and considering a number of 1,000 colonies, an efficiency of 10 to the 6 could have been reached. Chemocompetent cells prepared in the lab often reach an efficiency of 10 to the 6 or 10 to the 7, which is consistent with the estimation we made. Commercial competent cells can reach efficiencies of 10 to the 9. The reason why more colonies were observed for the chemocompetent cells could be that the bacterial suspension for preparing electrocompetent cells was not totally dissolved, and some of them could have died by the rising tension during the electric pulse. A negative control was not plated to verify the integrity of the antibiotic employed, but it would have been useful to determine whether the observed growth is triggered by successful transformation events or by a degraded antibiotic. According to the estimated efficiencies, the bacteria should be useful for non-complicated DNA transformations, like supercoiled plasmids. As the double strand of DNA is loose in a ligation procedure, the chances of incorporating a larger molecule decrease the transformation efficiency, and bacterial suspensions of 10 to the 6th efficiency or lower are not adequate to use. Electroporation proves a better procedure for these cases.
The main difference between the electroporation and thermal shock cell transformation methods is the way in which the DNA is directed into the cell. In the thermal shocks protocol, the DNA binds to the cellular surface, for which the electric charges of a chemically competent cell's membrane are neutralized. However, the electrocompetent cells are just prepared through the elimination of contaminants that may create electrical currents, since there is an electrical gradient that guides the DNA into the cell. The competent cells obtained through both methods in this practice showed high transformation efficiency due to the uncountable amount of cells observed in the LB with ampicillin medium.